all of that, and I'm still amazed I can find my shoes in the morning. I thought I would start with a very brief reading from my book, which sets the scene and explains why I set out on this journey and why the book is called Herding Hemingway's Cats. So this is from the introduction. You don't have to read along, it's fine. So it starts. It all started with a photo of a cat. I was hiding at the back of a scientific conference at the Royal Society in London when a cuddly-looking cat with unusually big feet caught my eye. "This is a Hemingway cat," said the lecturer, pointing at the animal on the large screen behind him. "They have 6 toes, they're polydactyl. Ernest Hemingway was said to be fond of them, and they still live on his estate in Florida today. And here," he poked at the computer, changing the slide to one covered with photos of misshapen human hands, "are polydactyl children with extra digits. It's the same genetic mistake that causes them." So looking at a 6-toed cat or a 6-fingered human, a natural assumption might be that it's due to a fault in a gene, but it's not. In fact, the cause lies in a faulty region of DNA that acts as a control switch, normally turning a gene on at the right time in the right place to direct the formation of fingers or toes as a baby or kitten grows in the womb. Not only that, but the switch is miles away in genetic terms from the gene it acts upon. Learning about the Hemingway cats and their broken switches, it got me thinking about my own understanding of how genes work and how I explain it to the public through my work as a broadcaster and writer. And so I wanted to write a book about how genes work. And so I travelled and Skyped my way around the world, talking to loads of scientists that I know. My background's in genetics. And I said, "I'm writing a book about how genes work. What can you tell me?" And pretty much all of them said, "Well, when you find out, let me know." Which really set the tone for the idea that there's so much we think we know about genetics and genes and how our genes build our bodies and make us us. But in fact, we barely know anything. And it is like herding cats, just trying to drag all these ideas together and understand what to make of them. So I thought I would present to you a little basic introduction to genetics. Has anyone got any kind of background in genetics, genes, DNA of any sort? Okay, you can skip the first half. And then I talk a bit about some of the stories that I uncovered in the book. And there's all kinds of tales in here. It's all first-person interviews. There's a few jokes, all sorts of things. So obviously, I'd recommend that you read it. But I thought I'd talk about a few of the stories that I uncovered along the way. So here we go. Da-da-da-da. Pity my whimsy. There we go. So obviously, you all work at Google. What is the internet made of? Cats. Science is also made of cats, but obviously with science cat jokes, you can't overdo them. It also turns out that DNA is made of cats as well. So this is DNA. You've probably all seen it. It's the sort of standard shorthand for, ooh, sciencey biology stuff. But this is the stuff of life, the instruction manual in all our cells. All of life has DNA. It's made up of 4 different letters, A, C, T, and G. These are 4 chemicals all strung together in all kinds of different orders. And it's the cell's instruction manual. It tells the cell to do things. This is a very basic cell. Well, it's an egg. But the best way to think about cells is that they're like kind of fried eggs, right? Stick with me, stick with me. The bit in the middle is the nucleus. This is where the DNA, not made of cats, the DNA made of DNA is in the nucleus, and the rest is called the cytoplasm. And all your body is made up of cells. You are made of trillions and trillions of cells. You have skin cells, you have brain cells, you have fat cells, you have toenail cells. We're all made of different cells, but they all have the same DNA in them. And it's the instructions in that DNA that make your cells do what they do and build your body. So this is the key thing to know. When people start talking about genes, and we hear about genes in the newspapers and stuff like that, people want to talk about a gene for alcoholism. Oh, it's a gene for Alzheimer's. It's the gene for cystic fibrosis. It's the gene for obesity. No, genes are things that make things in cells. They're basically cookery recipes. So when you're trying to talk about how our genes work, it's always important to remember genes are instructions that make things. And then it's how those things in our cells and our bodies and our brains are all put together and work that make us who we are, give us the abilities and skills that we have, and also the diseases that we get. So I'm going to do a really tiny bit of molecular biology, and I'm going to apologise because I'm really bad at PowerPoint, so please laugh appropriately. So I'm going to explain to you how genes work, and then we can all go home. Genes work like this. You have DNA, that's some DNA. That's a gene. So a gene is basically an instruction that is just a segment of DNA. It's kind of a recipe within a book. All the DNA in your cells is like a recipe book or a recipe library, and a gene is just one recipe. This is the thing that reads the recipe. It's called RNA polymerase. It reads the recipe. Da, 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 da. It took me so long to work out how to do that. I'm really bad at PowerPoint. I may even move on to like a star fade at some point. And it makes a molecule called RNA. This RNA polymerase makes RNA. And that is the message from the gene that then a cell uses to build something. This is the chef that's making stuff in the cells from the copied instructions in the recipe. It's called a ribosome. Reads it, makes something, and it makes protein. And proteins are the things in your cells that do stuff. So your skin is kind of hard and solid and keeps the outside out and the inside in because it's full of a protein called keratin. The cells in the back of your eyeballs can see because they have proteins that can sense light and can send those messages to your brain. All of our bodies are made up of proteins, kind of other molecules as well. But genes are the instructions that tell your cells to make proteins. They're recipes. So again, just to reiterate, genes are not things that give you diseases or give you traits. They make stuff. So a few facts, because I know you all like facts here, factual things. I was told to keep it nice and factual. My favourite fact about biology, you have 2.2 metres of DNA in every single cell in your body. This is absolutely staggering. I'm 5 feet tall. 2 metres is enormous. It's very thin, but it's 2 metres. I mean, come on, who's slightly boggled by that? Every single cell in your body, you have enough DNA in your body to go to the moon and back 1,500 times. I find this staggering. More people should, I'm a biologist, I find this stuff cool. Within that, you have 3 billion letters of data, effectively, of the A's, C's, T's, and G's, the instructions. It's a huge amount of data. And within that are the instructions, the genes, that tell your cells what to do. So how many have we got? Well, it was originally thought that we had, something upwards of 100,000 genes to make a human. God, we're probably going to need a lot of genes, aren't we? Because we're kind of cool. Turns out we've got about 20,000 genes. We're clocking in somewhere around fruit flies and nematode worms. So this was a bit of a shock. When they sequenced the human genome, they were like, what? Where are all the genes? They did a bet. They actually did this sweepstake to make people bet on how many genes there were going to be. And people started out like, 150,000, 100,000, and they're like, whoa. We basically don't really have that many genes. We use the ones we have in very interesting ways, which sort of proves it's not what you've got, it's what you do with it that counts. But we only have 20,000 genes or so. And all of that makes you you. And in fact, the sum total of all your genome, those phone books full of information that is actual genes, is really not very much. I told you I really wasn't very good at this. Only about 2% of your genome is actual honest-to-God genes. So this is the question, like, what's the rest? Any guesses? Cats. No, not cats. Good callback, though. The rest, people say, the rest is just junk DNA. It's just junk, it's just there. Is it just junk? No, it's not. So there are all kinds of things in your genome in between the genes that are, some of it is junk, true, but there are things like all the control switches that turn your genes on and off in the right time and in the right place to make you you and to make you work. There are genes, as well, I said at the beginning, there are genes that encode the instructions to make proteins, but there's also lots and lots of genes that are just read into that message, that RNA, and that does stuff in cells as well. There's a lot we really don't understand about that, but it's a very interesting area. A huge chunk of your genome, and I'm sorry to say this, is just dull and repetitive. Same as the actual work of sequencing the human genome is very dull and repetitive. Most of your genome is made up of things called transposons, or repetitive elements. These are long dead viruses or jumping genes that somehow got into our genomes, got copied and copied and copied and copied and copied and copied. Your genome is basically, a lot of the time, full of crap. And some of it is actively harmful. Some of it is garbage. We have some things in our genome that are really not good for us, and evolution is gradually clearing them out of the way. But a lot of the other stuff, the junk that's in our genome, people say, well, why have we got it in there? You know, everything must be here for a purpose. A lot of these people perhaps have a slightly religious agenda. And they cannot comprehend the idea that we can just have crap in our genomes that's just there. I'm going to do a quick survey. Hands up if you have some stuff in your house that is just there. Because the effort of getting rid of it, you know, if I had the world's most neat, clean, tidy office, that would be amazing. But the effort of actually tidying my office is unbelievably hard. It's much better just to put it all in a box and just leave it there. It's not hurting anyone. It's not in the way. If it starts to get in the way, or if it starts smelling, or, you know, I really need that box for something else, sure, I'll empty it. But it's the same with your genome. Evolution is not this perfect winnowing engine that gets rid of everything all the time it doesn't need. And I'm kind of sorry to break it to you, but you are not the pinnacle of our species. I mean, even though you work at Google, sorry guys, you are not the bestest, most fittest example of humanity that could possibly be. Evolution is a constant process, you know, so it's not perfect. And we keep stuff in our genomes that, you know, is just there. If it's not doing us any harm, it's just there. So I'm going to focus on some things in our genomes, the control switches, because there's some quite nice stories here that I cover in a couple of chapters of the book. Now, the control switches I'm going to talk about, these are the things that turn our genes on and off in the right time and in the right place to make the tissues of our body. And this is important because, as I said, all the cells in your body have the same DNA. They all have the same instruction manual. But the cells of your body are different. You have way more than 200 different types of cells. You know, hair cells, skin cells, fat cells, liver cells, kidney cells, all these kind of things. But you have the same instruction manual. So it obviously figures that you have to use your genes in different ways, in different types of cells to get the right outcome. It's the same thing as, you know, if you're making a glamorous dinner party, you'll want glamorous dinner party recipes, whereas if you're making a kids' party, you want kind of the kids' party recipes. So this is the problem. All our cells are not the same. And this goes back right to the very, very beginning of life. You know, you start life as one single cell, a fertilized egg when mummy and daddy love each other very much. And you grow into 2 cells, 4 cells, 8 cells, a little football of cells. You become a sort of little bubble of cells with a blob in the middle of it that is going to be you. And those cells are the stem cells that make a baby. And they have to specialize, they have to make decisions. They have to go, are you going to be head or tail? Are you going to be brain or skin? Are you going to be inside or outside? Are you going to be gut or liver? All the way down to the really finesse decisions. Are you going to be that bit of your fingernail or that bit of your fingernail? And all of this is determined by genes turning on and off at the right time and in the right place. So how does that work? Well, you can think of this a bit like a kind of a genetic switch, a control switch. So here's our representation of a gene, the blue thing with the arrow. People always draw genes like this. It's a gene, it gets read that way. That's why we draw them like that. The little purple thing near the beginning of the arrow, that's something called a promoter. Now, all genes have a promoter at the start. This is the thing that says, here's the gene. We start reading it here in that direction. Now, just having a promoter is not enough to turn a gene on. You need a molecule, a protein, a transcription factor to come and sit on that and say, okay, we're here, this is the start of the gene, we're going to do something here. But that's still not enough to turn a gene on. So what you have is a control switch, that's the little green thing there, and you have other molecules, depending on the type of cell that it is, that sit on those and say, right, we're going to turn this gene on here in our cells. And in other sorts of cells, they'll have a different suite of these factors that turn genes on in different places. And ta-da! That is enough to get a gene going. Now, obviously, that's just one thing. So that's only going to turn that gene on in the place where the green thing is happening. Does this all make sense? Yeah? Good. So, of course, if you want to turn genes on in, say, different tissues, if you need the same gene to be switched on in the brain, in the liver, in the kidneys, in the lungs, but not anywhere else, you need a little bank of switches that all respond to different factors that are there in that tissue at the right time. So that's that's about kind of a year and a half's worth of genetics and molecular biology course there, guys, so well done. And now we get to the fun stuff. This is Ernest Hemingway. I love Ernest Hemingway for many, many reasons, mainly because, God, that guy could write a sentence that punched you in the face. But Ernest Hemingway also liked several things. So things liked by Ernest Hemingway, I have done a Venn diagram. Ernest Hemingway likes fighting. We know from books like Death in the Afternoon, he likes fighting. He also likes fishing, mating. And all these things come together in cats. I knew I'd get back there in the end. So, and specifically these cats. Now, this is a polydactyl cat. Like I mentioned at the beginning, it has multiple toes. You can see its thumb there, bizarre three-clawed thing. So this is a polydactyl cat. And Ernest Hemingway had loads of these cats on his estate down in Florida, down in the Florida Keys. And the story goes that he was given one cat called Snowball, or possibly Snowflake, the legend is a bit uncertain, by an old ship's captain. Gave him this six-toed cat, and Hemingway kind of liked them because he likes the sea and sea captains, so he kept it. And the stories about why so many of the ships had six-toed cats, because they're very popular all down the eastern seaboard of the US. There's loads and loads of the cats have six toes. There's an idea that maybe they're better at catching mice. You know, you've got kind of big extra paws, toes, you can get them, important on a ship, useful. If the ship's kind of rocking about a lot and you have a prehensile thumb, you could grab onto a rope or something like that. You just imagine this cat like hanging onto the rigging, that kind of thing. There are many reasons why cats with six toes may be useful on board ships. The main reason, I think, is probably this one. Because, like, what's more lucky than a cat with five toes, right? So let's look in a little bit of depth at the switches that are responsible for this particular phenomenon. So this is the bit of proper genetics here. This is the part of the cat genome and also the human genome that is responsible for building toes. I mean, not specifically building toes, there isn't a toe gene, as discussed, but there is a molecule, and it's called Sonic Hedgehog, yes, after the cartoon character. That's that sort of little SHH thing where the green arrow is pointing. And that is responsible for basically saying, let's build some digits here. Let's do something here. Now, Sonic Hedgehog is used all over the body during development. It's used to build digits. It's used to make decisions in the brain. It's kind of a deciding molecule. It decides things for cells. So it gets turned on in the brain. Those are the blue things called CNS. Those are the central nervous system switches. It's involved in the skin and the guts, the epithelial linings. Those are kind of the orange switches. But the one I'm really interested in is the green one, and that is the limb bud switch, which turns on Sonic Hedgehog. That's the little blue patch there. This is like the developing paw of a mouse in the womb. It turns on Sonic Hedgehog in a little strand at the bottom where that's sort of the fleshy part of your hand's going to be. And it says, we're going to make some fingers, and these ones are going to be like pinky, fourth finger, third finger, all the way through to the thumb. It's sort of a gradient that says we're going to do fingers like this. So it's a really important switch. And obviously when it's faulty, you end up with the cells in the hand, in the developing hand, just kind of not knowing what to do. They just start making more and more toes or thumbs because they're just not really sure what's going on. And it's the same mutation in cats and a similar mutation in polydactyl humans with multiple fingers as well. So these switches are kind of really important. And obviously there's an evolutionary advantage to cats having 6 toes because, as I said, on ships they're perhaps better at grabbing on, or just that sailors favoured them. And who knows, maybe one day these cats with thumbs may gain other skills and possibly learn to kill us as we sleep. And I, for one, welcome our feline overlords. But there's an interesting and more important thing here. So these switches in the genome are evolution's playground. So we think about evolution as very small changes. You know, we're taught at school that evolution is very, very slow. At one change and you get a small thing, you know, maybe a tail gets a bit bigger or a neck gets a bit bigger, or, you know, you get a bit more spotty or something. Evolution is very slow and gradual. But as we've seen, just one faulty switch is the difference between having 5 toes and 6 toes. So small changes in these switches can make really big changes to organisms. And I'm going to tell you about one of these stories now. So I'd like you to meet some fish. We go from cats to fish. And these are sticklebacks. Anyone seen sticklebacks, gone fishing for sticklebacks? They're fish, they're spiny. So the top ones are sticklebacks that live in marine environments. They live in the sea. Now, about 10,000 years ago, there were lots of sticklebacks like that, living in the sea, and they would always swim up the rivers to spawn and do whatever it is sticklebacks do. I mean, probably carefully, if you've got those spines, I'll tell you. So they swim up the streams to go into fresh water to spawn, and then they come back down to the sea. Now, a bunch of sticklebacks came really unstuck in the last ice age, in that they got up stream and then they got stuck. So you separated 2 populations of sticklebacks. So you've got the ones on the top that are marine dwellers, and over the course of 10,000 years, the sticklebacks in the freshwater lakes changed to be like the ones below. Now, can anyone see the difference between those 2? Slightly bigger fins, yeah? The key thing to notice is the thing that looks a bit like a willy on the top one, kind of poking down. It's not a willy, because I wouldn't show you that. In fact, well, that's coming later. But that is some pelvic spikes. So the sticklebacks that live in the sea, they have these spiny fins to fight off predators. It's something like kind of fembot, like pew, pew, pew kind of thing. They have these spiny fins to keep them safe, because there's loads of predators in the sea. The sticklebacks that ended up in the freshwater lakes didn't have the same predators, and they don't have these spiny fins. So a guy in Stanford in the US called David Kingsley got very interested in this, and he said, well, what's the difference between these 2 fish? Because to all intents and purposes, they're only separated by 10,000 years. That's not very long in evolutionary terms, and they kind of look pretty similar. So here's an X-ray of these fish. So the top one is the marine sticklebacks, and the bottom one is the lake-dwelling freshwater sticklebacks. They've got no bloody pelvis. Their entire pelvis is missing. So not just they haven't got the spiny fins, or their spiny fins are a bit smaller or anything, the whole thing's just gone. That is pretty radical. And for that to happen in just 10,000 years, which is like it's nothing in evolutionary terms, that is amazing. And it's down to one broken control switch in a gene called PITX1. Don't really need to know what it does or what it's for, but one single change in a control switch that turns the gene on at the right time to build the pelvis in the sea sticklebacks. Broken in the freshwater sticklebacks, no pelvis at all, gone, vanished, gone. It's absolutely incredible. And what's even more amazing is that you can take that bit, that control switch in the DNA, and you can put it back into freshwater sticklebacks with no pelvis, and they will grow one. So you can see there that little, that shouldn't have that bit there. They've put that control switch in there. It turns the gene back on at the right time, and these fish, first fish of their type for 10,000 years, growing a pelvis and like their spike things. Who knows what they thought of that? Maybe they wake up and like, whoa, whoa. And this is not the only example. So David Kingsley's looked at other examples. For example, we have a gene called KIT-LG. This is responsible for things like pigmentation in the hair, in the skin. You get two different versions of a control switch. There's one letter difference in this control switch by the KIT-LG gene. One of them gives you brunette hair. The other gives you blonde hair. That's one single letter is the difference. Now, I mean, I grew up in the 80s, and I would make a lot of blonde jokes. And I'm sorry, I didn't know any better then. But, you know, I know now. But it's one letter that's different. And, you know, blonde jokes aside, there are more fundamental things. Again, there's another control switch. One single letter difference is the difference between white skin and darker skin. Now, it's not like an on-off. If you've got this, you're going to be white. If you've got this, you're going to be dark. Because, you know, we are more than 50 shades of people here. But it's kind of turning the dials. It's ramping genes up and down, making them more active. You make more pigment or less pigment. And it's one letter in the 3 billion letters of your genome. Now, as I was researching this and kind of writing about all of this, it was just when all the race riots were going on in the US, you know, the police sort of shooting unarmed black people. And I sort of sat there and I thought, this is ridiculous. It's one letter in our DNA, and people have died for thousands and thousands of years because of this. So anyway, that's the philosophy bit. And now here comes the rude bit. So I don't know, ladies, if you've noticed, but if you've ever had sex with a chimp, they don't call you in the morning. There are certain species, mammal species, things like chimps, mice, tomcats. They have what's probably nicest to call it, a promiscuous lifestyle. They sort of prefer the wham bam, thank you ma'am kind of school of procreation rather than anything else. And they certainly, you know, don't spend their Saturday mornings in IKEA. I'm just about to get divorced, so I don't have to spend my Saturday mornings in IKEA anyway. Yay! But what is different between these species and human males is if you look in the trouser department. I think this is going on the internet, so I probably need to be a bit euphemistic here. But if you look on the genital equipment of things like the tomcat, the mouse, and the chimp there from left to right across the top, they have these kind of spikes on their penis. That's a technical term there. Is this going to be explicit content? It's fine, okay. So they have what's called penile spines. And these are, for some reason, meant to like grip on, you know, if the lady's running away going, you never even bought me dinner, that kind of thing. They're meant to grip on and enable the males to go for their sort of fast and furious mating style. Guys, I'm assuming that you guys don't. If you do, you should go and see a doctor. And some human males do have like little nodules, and they're the remnants of these. But the key difference here is that there is a single genetic switch that is different between all the species with spiky penises and human males. and it's a switch that controls a gene called the androgen receptor, which is a receptor for the male sex hormone testosterone. You know, it makes you grow your male characteristics and things like that. And this one change in this switch is responsible for this difference. So, you know, you can tell a lot of evolutionary just so stories about all these kinds of things. One is maybe that some lucky proto-human developed this in his genitals, and the ladies that he was going with were like, oh yeah, I think I prefer that to the the spiky one. Do you want to do it again? And maybe that encouraged pair bonding and the more sort of monogamous social relationships that humans have. I don't know, am I right, ladies? I personally don't really fancy a spiky boner. Don't know about you. So those are some stories about the control switches in our genomes and how they can make some really big differences to the outcome of an organism. The other thing I want to really briefly touch on before I finish is that I've drawn a lot of pictures for you of the idea of a gene as this long straight line. You know, we've got a gene, we've got the switches and things kind of sit on it and it does this stuff and it's all very linear. But as I said right at the beginning, you have 2 meters of DNA in every single cell of your body. That's 2 meters of DNA in something that's less than the size of the head of a pin. So biology doesn't look like these kind of nice, neat diagrams. The DNA in your cells kind of looks like this, but much, much more messy and compact, and it's wrapped up around all sorts of proteins to keep it packed in there. And you're like, wow, how does anything find anything in there? And the idea is that, you know, our biology is actually not as neat and linear as you might think. You know, you can see a diagram like the one on the top and think, okay, you know, these factors find these genes and they turn them on. Stuff's got to find each other in this complete mess. And sometimes it'll work and sometimes it won't, and genes will go on and genes will go off, and you have to bring all this mess together. And I touch on it a lot more in the book, and I'd really encourage you to find out a bit more. But we are products of probability. This is a stochastic event, or random, as I prefer to term it. Scientists prefer stochastic because for some reason just saying like life's kind of random upsets people. This is a stochastic process. Genes aren't exactly switched on and off. They're more like kind of dials, suggestions, landing platforms for the things that make them work. And with that in mind, I'll tell you a final story before I finish, which is about the wobbly worms. So there's a chap called Ben Lehner who works in Barcelona, and he works with these tiny organisms. These are nematode worms, C. elegans. They're 1 millimeter long. And loads of biologists love them because they are completely stereotyped. They do exactly the same thing at the same time. They have an exact number of cells. It's about 1,000 cells. And they all divide in a completely programmatic way. So you can start with one fertilized nematode worm egg cell, and you can watch on cue as the cells pop into existence and divide and specialize. And they all have names. Every single cell in that worm has a name. They're called things like AB1 and stuff rather than like, you know, Carrie and Doris. But every single cell in that worm has a name. And the worms that he uses are genetically identical. They're like super clones. They're all exactly the same. And so you would assume that if you damaged a gene or a control switch or something like that in one of these worms, it would come out with exactly the same outcome all the time. And this is where it gets weird. Because you have this gene, you make a mistake in it, half of the worms die. Now that's weird, because they've all got the mistake in that gene, but only half of them have died. So that tells you that there's a whole bunch of buffering going on in the genome. And, you know, like I say, this is a stochastic event. This is not a yes-no. And it highlights that our genes are not yes-no, deterministic, on, off, make this, don't make that. There's a lot of randomness in there. Interestingly, if you break 2 genes, you can shift the balance. You'll end up with 10 alive and 90 dead if you look at 100 worms that all carry this gene fault. And and if you start breaking more parts of the system, you'll end up with all dead. Biology is incredibly robust. We wouldn't be alive if it wasn't. And the final thought I will leave you with is this, is that the more we start delving into our genomes, the more we sequence, the more we understand. We're now at this point where we kind of sequence all the things. We're starting to sequence more and more human genomes. And we're discovering incredible stuff. We're discovering the fact that each one of us is walking around with about 30 genetic faults that should probably kill us. But they don't. Because you've got all this other stuff that's going on and buffering in your genome. And that's what keeps us alive, and that's amazing. Your genome is amazing. I hope you enjoy it, and I hope you also enjoy my book.
Lovely, thank you, Kat. We've got time for a few questions, if anyone has questions. Whilst you think of one, I will kick off. With the humans, we have about 20,000 genomes, and you said we're and we're quite inefficient in terms of like the baggage that we store and so forth.
Yeah.
So does that mean animals which have a much faster, shorter lifespan, fruit flies and so forth, are they more efficient?
Yeah, broadly. So fruit flies have smaller genomes. They sort of carry less crap with them. It is like the idea if you live in a family home for like 60 years, you will accumulate loads of rubbish. It's broadly that kind of law. And one of the most incredible genomes is the pufferfish, fugu, the sort of Japanese delicacy. And that's got pretty much all the same genes that we have, but an eighth as much DNA. So that's really interesting. And when people go, oh yeah, we need junk DNA, we need it for all these things. I'm like, well, you don't need it pretty much to successfully build a vertebrate. So it's an argument that's still running.
Very good, all right. Yes. And if you could repeat the question.
Oh, okay, sure.
How do you even count the genes? How do you know what isn't a gene, where they start, are they in, that they detonate isn't other genes?
That's a great question. So how do we know what a gene actually looks like in the genome? So this is the job of something called genome annotation, and there's all these people at the Sanger Centre in Cambridge and the EMBL and things like this doing it. So remember I said that there's a promoter at the start of a gene. That's a really characteristic DNA sequence. You can spot it. There's things like something called a tatter box, and that's a run of TA, that kind of things. So you can spot those. I don't want to go in too much into the molecular biology, but you get little groups of 3 DNA letters that make the individual building blocks of a protein. So if you get a long, uninterrupted string of things that you know are making proteins, you kind of go, okay, that's probably a gene. There are some other characteristics as well, but this is where it gets hard because there's been this sort of arbitrary cutoff for how big a gene is. So you go, okay, it has to be over a certain size, it has to make a protein that's at least this big to be a gene. And now we're discovering that there's all these kind of tiny little genes that actually make proteins. They're called SMORPs, small open reading frames. And it's just because we didn't just arbitrarily said, oh, those can't be genes. So, it's like a fishing net. If you only catch the size of things that you think you're fishing for. And again, we're saying, as humans, these are genes. And in fact, there's a lot probably that we don't know that's doing stuff in the genome. So it's an open question about what's actually useful and doing stuff in the genome. Hiya.
So, my understanding is that when we're sequencing genomes, we break everything up into little bits, and then we count all the little bits, and we go, oh, they probably go together in this order as a rough. How likely is it that something will come along and we've been doing sequencing wrong, or how statistical are our measurements, I guess is the question.
Oh, so this is how accurate is genome sequencing if we're just kind of ripping everything up and sticking it back together again. You're looking for overlap. I mean, now the next generation sequencing. So when I was first a scientist, the way we sequenced genomes was you'd sort of start here and read a bit, and then you'd start here and you'd read a bit further. So it was really, you kind of knew what you were patching together. You'd look for really big overlapping bits of DNA. The new generation of sequencing kind of takes everything up. It's like taking a book, ripping it all to pieces, reading it all at once and trying to assemble the words. And again, you're looking for multiple overlapping regions. And yeah, it does come down to how good your algorithms are. And this is where the problem is that so much of the genome is repetitive. That has been ignored. And now people are starting to talk about, the platinum genome. I heard a lovely analogy. Ewan Burney, who's one of the big sequencing guys, he said, for ages we've drawn this map of Europe that's like the genome, and everyone's gone, oh God, I don't want to have to do the fjords. But, but now we're having to do the fjords because there's probably interesting stuff in there. Or we need to know what's in there so we know it's not interesting.
So given the fact that we have relatively fewer genes, but the total size of the genome is comparable to, and probably on the larger end, does that mean that the average size of our proteins is typically bigger than those of other animals?
Well, no, we've basically got mammalian genes. So, there's a pretty standard set of genes and proteins that build vertebrates, build mammals. And our genes are broadly the same as those. There's nothing in our genes that's particular to that. It is in the junk, in the control stuff, in all the other stuff that is special to us. And it's quite an interesting thing to get your head round, because obviously we basically share all our genes with a chimp. We are pretty much 99.9% chimp. So the difference between us and a chimp must be that our control switches are human control switches in our rest of our human DNA, whereas a chimp's got chimp control switches. So you have primate genes plus human switches makes human, primate genes plus chimp switches makes chimp. So the suite of genes is very, very similar, but it's all the other stuff that's different.
So it's been quite a lot of studying the genes, although there's been cloning and all companies that offer to read your genes and DNAs. But I was wondering, at this point, how far is science of actually being able to play around with the genetic material? It's
Okay, in terms of editing and design, is this the design a baby kind of question? How close are we to taking that information and manipulating it, or just understanding
or even if you get to know your genetic material, is there anything you can do about it at this point?
Yeah, sure. The answer broadly is no. I need to be careful because Google own 23andMe, don't they? A tiny bit. So there's lots and lots of organisations now that will sequence your genome, or at least look at bits of it and say, okay, you've got this which is linked to this tiny increase in risk of X, Y, Z. There are a few diseases that we know of, things like cystic fibrosis is a great example, that we call the Mendelian diseases. It's one gene, one fault in it gives you the disease. The faulty gene in cystic fibrosis, it makes a protein that kind of shuffles salt in and out of your cells so you don't make mucus properly and all this kind of thing. Most diseases, things like Alzheimer's, obesity, characteristics like intelligence, even things like hair colour, skin colour, weight, all sorts of things. Many, many, many genes. And it's about variation, things being dialed up and down. And like I showed you with it, stuff's just kind of a bit random as well. There's a whole random element in there. And that's what's been really shocking about doing the sequencing studies and discovering that lots and lots of people are carrying mutations that should be manifesting as a disease, but aren't. So this is a problem. If you have your genome sequenced, we're starting to go, well, we thought that if you had this, you would be like that. But now it's well, if you have this, you might be like that. It might depend on 1,000 other variations in your genome, and we don't understand all that interplay. So there's a huge, I would recommend you read my book, because there's a huge chunk in there about how people are trying to unpick this and trying to understand how we integrate all the information in the genome, not just one gene. And this is where it comes on to the sort of designer baby genetic manipulation question, because there's all these techniques you might read about in the papers called CRISPR and things like that, where you can cut up bits of DNA and manipulate them and chop stuff around. For some characteristics, for maybe these one gene, one disease kind of characteristics, I think that could be incredible technology, real life-changing technology for families that are affected. Whether you're saying, oh, can I do a bit of chopping up and make a more intelligent baby? there's so much we don't know about the genetics of intelligence and the huge chunk of environment, lifestyle, upbringing that comes on top of that. So I think engineering more complex traits is going to be incredibly hard, and you're probably better off just having sex with a clever guy.
In terms of the nature-nurture argument, my understanding of epigenetics, which is very basic, is that it's those, you can pass the grandparents' genes far through to the grandchildren. If the grandparents were obese, that might get passed down.
Okay, yeah.
Is that something that is, where's that at in terms of science?
So in terms of where the science is with that, it's a little bit shaky. So the idea of epigenetics is a very, very misused and incredibly abused phrase. It's so abused that the poor thing needs therapy. Epigenetics is basically describing how your genes are used differently in all the cells of your body. You know, like I said, you've got the same DNA in all your cells. You need to turn genes on and off. That's epigenetic. It's in addition to the DNA, to the genetic code. So that's fundamentally what epigenetics is. The kind of stuff that the papers get very excited about is the idea of transgenerational epigenetics. There's something that happens in the DNA of a grandparent could get passed on to their egg or sperm cells and then into the next generation. And this isn't changes in the DNA, this isn't mutations in the DNA, this is the sort of information about stuff that's happened in a lifetime that has affected how the genes work, that then get passed on down the generations. And in humans, the evidence for a lot of this is sketchy. It probably does happen, but not in the way that people think. And there's been a couple of really exciting papers just in the past couple of months suggesting that maybe tiny little fragments of RNA might be responsible for transmitting this information. But it's a very, very new field. And actually, when I went to talk to all the researchers I was talking to, I asked them, you know, what's cool, what's interesting. And then I said, last of all, what's weird? You know, what makes you think, evolution, go home, you're drunk? And so many of them said transgenerational epigenetics. It's like, it's really the frontier about how your lifestyle, things you do in your lifetime, can they get passed on to the next generation and the one after that, and potentially even further. So, and there's a whole chapter about it in the end of my book. Read my book.
So this is maybe a bit related. How much information about creating a human is in the DNA compared to in sort of transient in living humans? Or if I had just human DNA, would I need a human egg to make a human? Or if I had like T-Rex DNA, would I need a T-Rex egg to make a T-Rex?
That is a really great question. If you had human DNA and you put it into, you know, like a primate egg or anything, into anything. The problem is that life is a kind of a slippery concept. So life is not just the molecules. Life is not just DNA. All cells, all living things come from existing cells. So you would definitely need to take the DNA and put it into something. I mean, this is what Craig Venter is trying to do, and he's doing it with like very simple bacteria, synthesizing DNA and putting it into a hollow cell. And everyone goes, oh my God, he's created life, we're playing God. It's like, he still took some DNA and put it into effectively a living cell. In terms of whether the switches and stuff would all work, some of them probably would, some of them probably wouldn't. You can take, say, a human chromosome and you can put it into mouse cells and it will start working fine. And there's a really interesting model of, I think, Down syndrome that's based on putting a human chromosome into mouse cells. So yeah, some of it would probably work. I don't know. I've got time, I'll write a grant. I'll let you know.
Good. Gentlemen over there.
Hi, you mentioned that we all carry around roughly 30 mutations, and also that our shared DNA with, say, a chimp is incredibly similar, and the only real difference is the control switches. Roughly how many of those control switches would have to go wrong for someone to give birth to a chimp?
Also a fantastic question. How many control switches would we, how many control switches in the genome would we have to change to turn a human into a chimp? Probably Not that many. Well, and we are extremely similar, and it's probably a few of the key developmental genes. So some of the brain ones, the obviously like the hair and face thing, the bony penis business. Actually, there's a really interesting story that I had to take out of the book about this woman in Stanford who's discovered that a lot of the switches responsible for the shape of human faces compared to chimp faces are derived from long dead viruses in our DNA. So it's like literally these viruses are the things that have made us humans compared to chimps. So I reckon if you started knocking out loads of those, you would start to get back towards something that looked more like a chimp and less like a human. Must publish that at some point. But yeah.
Yeah. We'll take one more question. Gentleman behind there. That's fine.
Just going back to the Mendelian diseases, which I assume is your repressive dominant gene stuff we learned at GCSE, 1 in 4 chance of parents, blah, blah, blah. What makes it easy or not easy to track down the gene responsible for them? I mean, we've got cystic fibrosis and sickle cell edema. Is it just if you get enough sample sizes of people with it and without it, you can find it the same, or some diseases are fundamentally harder than others?
Yeah, so a lot of the genes that we've known about for a long time, they were really done the hard way by tracking pedigrees, but it's still the same about taking sections of people's DNA and reading it and comparing, and there's sort of various mapping techniques that you can do. So all through the 80s and 90s, people were sort of homing in on regions by looking for regions that were similar between people and regions that were different, and going, okay, all these people with this disease have broadly this area that's all similar, so it's probably in there. And it took years and years and years and years and years and years. And then with the sequencing revolution, all you do is you take someone who's got it, take someone who hasn't, or a bunch of people who've got it and a bunch of people who haven't, and sequence them enough, and you'll probably find some differences. And then you, because of what we know about what the genome looks like, we have quite a good map. You can go, okay, if this is a syndrome that's involved in the shape of the face, perhaps it's a gene that's involved in like cartilage growth or something like that. So you can make some pretty sensible deductions. What gets more challenging is when you're looking for the more subtle variations. So I work for Cancer Research UK, and we do a lot of these studies on cancer risk. And you have to take tens of thousands of people and look for these tiny variations between them, and take 40,000 people with cancer, 40,000 people without cancer, and it's got to be the same type of cancer, and say, okay, broadly, what marries up between having it and not having it? And it's a risk thing. So it's starting to get very hard to find these variations and to find new genes, and people are doing bigger and bigger and bigger studies and finding less and less and less. And I think there's probably limited benefit to be found from keeping pursuing with those kinds of studies personally.
Lovely. Well, we'll leave it there. Please join me in thanking Dr. Kat Arney.